Hey everyone! In this video, I'm going to be going over Chargaff's rules and the nitrogenous bases. You're going to be able to describe nitrogenous base pairing by the end of this video, as well as use Chargaff's rules to distinguish between single-stranded nucleic acids and double-stranded nucleic acids. Finally, we're going to describe how the size of purines and pyrimidines affect the width of double-stranded nucleotides. So let's get started. Now, Chargaff is a scientist who discovered that the ratio of adenine and thymine were almost always equal or approximately the same in organisms, as well as cytosine and guanine were relatively the same in their proportions. So the pairing typically goes, adenine pairs up with thymine and cytosine pairs up with guanine. Now remember that those nitrogenous bases are found at carbon number 1. So this line right here actually is not a bond between the two nitrogenous bases, but rather a hydrogen bond, which is not a true bond. There's no sharing of electrons between these. It's more attraction between the bases. And between adenine and thymine, there are going to be 2 hydrogen bonds. And between cytosine and guanine, there will be 3 hydrogen bonds. So that means that it's hydrogen bonds that keep the 2 strands of the nucleic acid together. If this was an RNA, you would see uracil here instead of thymine. So once again, for double-stranded nucleotides or double-stranded nucleic acids, it's the hydrogen bonds that keep the. So we can use Chargaff's rules to determine whether or not an organism is double-stranded or an organism has double-stranded DNA, or in the case of viruses, this could be single-stranded DNA, or it could be single or double-stranded RNA. So let's see how you would distinguish between these groups. So the one that most of you are probably familiar with is double-stranded DNA. So how are you going to know by looking at an organism's genome or looking at the base pairs? How would you know if this organism had double-stranded DNA? Or this could be a virus that has double-stranded DNA. So the first thing you want to look for is thymine present? If thymine is present, then that means you are looking at DNA. The other thing you're going to want to look for to see if this is double-stranded DNA is that your ratio of A to T will be relatively equal, and your ratio of C to G will also be relatively equal. Let's say you're investigating a virus now, and the virus is suspected to have single-stranded DNA as its genome. How would you know whether or not this virus has single-stranded DNA as its genome? Well, number 1, again, you would look for the presence of thymine. The big difference is here. If it's single-stranded, then you do not expect the ratios of A to match the ratios of T. In fact, these numbers could be wildly different. And C is not going to match the percent of G. Now, some viruses have double-stranded RNA as their genetic information. And the thing about RNA that you need to remember is that RNA does not have thymine. So what you would see is similar to what we have with the double-stranded DNA, only now thymine is absent, and instead you're going to have uracil present. But the ratios of A to U are going to be very similar, and the ratios of C and G will also be similar. So last is the single-stranded RNA. And like the single-stranded DNA, those ratios of A and T don't match, and C and G are not going to match either. But again, uracil will be present. Now, the nitrogenous bases can be classified as a purine or a pyrimidine. So the purines are large and have 2 rings of carbon, and the nitrogenous bases that are purines are A and G. The pyrimidines are small and have 1 ring of carbon, and the pyrimidines are TCU. And so it's thanks to this pairing of 1 purine and 1 pyrimidine, in other words, A and T, or G and C, that allow DNA to have the proper width when it is double-stranded. So again, it is 1 purine and 1 pyrimidine that are going to be side by side here where the nitrogenous bases This would be the sugar phosphate backbone, but here would be the hydrogen bonds between the nitrogenous base pairs. And so if you have one purine and one pyrimidine, the DNA has a certain width apart. Stop and think for a moment. What would happen if you had a mutation where you had two purines side by side? Make a prediction. Okay, let's see if you predicted correctly. So instead of having the proper width, at the point where the two purines would be side by side, the DNA would have a bulge in it, and it would look kind of something like this. So because the two purines are very large, they would take up more space in between the nitrogenous base pairs, and so you'd see a bulge in the DNA. Now, what happened if there was a mutation where now you have two pyrimidines across from each other? Take a moment right now to make that prediction. Okay, so hopefully what you predicted is instead the opposite, where now the DNA would actually kind of cave in a little bit because the purines are very small. The width of the DNA at that point would be much smaller compared to what it should be here. Now, it's these nitrogenous bases that give organisms their unique genetic code. So we think of DNA as a fingerprint and that your DNA is unique compared to any other person in the world. But it's not that sugar phosphate backbone. It is those nitrogenous bases, the purines and the pyrimidines, that we can use almost like a fingerprint. It's unique to each species. So even though you may have differences between my DNA and your DNA, for example, overall, humans have certain ratios in the amount of A's and T's we have. And if you compare this to a fern or to a potato or to any other organism, it's those nitrogenous bases that are going to be in different ratios that allow for each species to have a different genetic identity. Okay, so in this video, we described the nitrogenous base pairs, A with T, C with G, and the number of hydrogen bonding that occurs between them. We used Chargaff rules to distinguish between single-stranded nucleic acids and double-stranded nucleic acids. Chargaff's rules would help us identify the difference between single-stranded versus double-stranded nucleic acid. And we described how the size of the purines and pyrimidine affects the width of double-stranded nucleic acids and how mutations would then affect that width. I hope this video was helpful. Thanks for watching. Bye.